happening in the mitochondria. The ammonia is freely floating around because we cannot let it float around in the bloodstream. It's too toxic. So we need to let it be carried by something, some intermediate. And the intermediate is, in this case, carbamate. Yeah? Okay? So carbamate, okay, to carbamoyl phosphate. Catalyzed by the enzyme carbamoyl phosphate synthetase 1. Difference is synthetase 1 is found in mitochondria. First difference. Synthetase 2 is found in cytosol. Alright, and the nitrogen donor in the synthetase 1 is ammonia. Free ammonia. Right? Free ammonia. But in the previous reaction, nitrogen donor is glutamine. Okay? Glutamine. Two differences. Alright? So the two isoforms, although sound similar, although catalyze the same reaction with bicarb, they are located differently. And the donor is different. Okay? So over here, but the product is the same. Product is the same. Carbamoyl phosphate. Same product. Okay? Carbamoyl phosphate. Yeah, two different pools. This is the cytosolic pool, that one is the mitochondrial pool. And over here, the carbamoyl phosphate is then condensed, right? Okay, to add. So we already have glutamine, we have bicarb. The next precursor is your aspartate, right? And in this case, aspartate again is added wholesale. Yeah, but because every part of the skeleton is needed, we don't remove it as fumarate. Because we need all the parts of the skeleton, not just the nitrogen. So in this case, the carbamoyl phosphate condenses with aspartate to give carbamoyl aspartate. Okay, so phosphate group is always an intermediate. It gives it this high energy, but in the end it's always removed, and the whole moiety gets added on to the phosphate part of it. Okay, this is catalyzed by aspartate transcarbamylase or ATPase. Okay, so you see the aspartate now gets added. Pyrimidine is so much easier. The reaction is so much easier. And then look at here. What do we need? 1, 2, 3, 4, 5, 6. We already have the 6, right? 6 atoms in the structure. So we just need somehow to close this up, right? We need to close this up. We need to add a ribose. We can complete the process. All right. So indeed, we do condensation. Okay. Again, same reaction. The ring closure is by condensation. Okay. And this enzyme is called dihydrooroutase. This structure is called an oroutase. Right. It has the extra CO minus group. Okay, this is not needed because just now you saw that the atoms that we need is this, 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 this. So if this is extra, how do we remove it? COO, so convenient. CO2, right? It's CO2. So once, okay, it closes up by this enzyme called dihydrooroutase. Okay, it becomes a dihydroorotate. Okay, hydroorotate or oroutase. And now the carbon dioxide. Okay. So over here, okay, the dihydroorotate is first oxidized. So over here, they are all saturated bonds, yeah. And now you need to saturate this bond in the final ring structure. And this enzyme is dihydroorotate dehydrogenase. Again, ring a bell, dehydrogenase. Okay, whenever we see dehydrogenase, we know coenzymes are involved. Whether NAD or FAD, that one is hard to guess, so must memorize. So over here, but at least we know that electron carrier is involved. And most of them, if they involve flavin, okay, this one involves flavin, FMN, and iron. If they involve flavin, then you know where they are. They are not floating in the cytosol, they are located on the outer surface of the inner mitochondrial membrane. We have seen this in TCA cycle. Okay? So again, once you see flavin, automatic, inner membrane of mitochondria. Okay? On the outer surface. Right? Why? Why is it associated in the inner membrane? because it's linked to the electron transport chain. It has to be closed. If it's floating in the cytosol, it cannot pass on the electrons. Right? And we know that it passes on to quinone, one of the electron carriers in the complex, respiratory complex. Right? So this is the only enzyme, the dehydrogenase, that is in the mitochondria. The rest of the enzymes for this process is cytosolic. Okay, cytosolic. I don't know why this is missing. Okay, so cytosolic, and this gives rise to oxidation process. Oxidation process, single bond changes to double bond, hydrogen is lost. Okay, hydrogen is lost in the form of electrons. Again, same concept. Okay, as TCA cycle. Hydrogen is lost in the form of electrons being carried by your FMN, which will pass it on to quinone. So you get a molecule called orotate. So dihydroorotate, this is saturated with lots of hydrogen. Orotate is saturated over here. Okay? Okay, now this ring is complete, but we don't have a ribose. And how do we add ribose? Same, you go back to the purine process, ribose is added in the form of PRPP. This is what I meant, okay? In purine, we started with ribose first. But over here, we formed the ring, then we added on to the ribose in the form of PRPP. Okay? Alright? So this is catalyzed by the enzyme phosphoribosyltransferase transferase. Just replace the substrate, okay? Orotate. Right? And you will get a monophosphate. Orotidine, 5 monophosphate, 5 position. 5 prime is the phosphate position. Okay? Yeah, monophosphate. And finally, the only problem here now would be this carbon dioxide. We need to remove this carbon dioxide. Already it looks very similar to uridine, to U. So the final reaction is the removal of this COO minus in a decarboxylase reaction. Okay, OMP decarboxylase. Okay, and then carbon dioxide removed, and you will have your uridine monophosphate in this reaction. Complete. Okay? Alright? So this UMP can be converted to CMP by glutamine. Okay, over here, you just add an NH2. I didn't go through in this reaction, but can be done. Okay? Right? By glutamine addition over here. Okay? So far what we have achieved is IMP and UMP, is a monophosphate. But we know that the trisephosphates are the major reactive components. So how do we make it? Okay, so initially when I said the key concept is kinases will convert it to triphosphate. So monophosphate to triphosphate is an addition of phosphate group process. So kinases are involved. Right? So that is a stepwise reaction. First, you can add AMP to become ADP. But it's very interesting that in order to add phosphate group, we don't start from inorganic phosphate. In fact, we need the help of ATP to help us add extra, okay, phosphate group to the AMP. Right? Okay? So ATP is in large excess, hopefully. It should be in large excess in your body. Alright? And very reactive. Therefore, we use ATP. Much more reactive than inorganic phosphate. So in this case, in order to make AMP ADP, that means you add one more phosphate group. You have this enzyme called adenylate kinase, okay, to catalyze this reaction. ATP to AMP will give you 2 molecules of ADP. And this ADP doesn't go any further, okay, by kinase reaction. It has to be phosphorylated to ATP either by substrate level or by oxidative phosphorylation. Okay, this is how it works. ADP becomes ATP either by substrate level or oxidative phosphorylation in the body. Right? We have seen this oxidative phosphorylation, the F0, F1 ATPase, okay, reaction. Okay, so these are called nucleoside monophosphate kinases. This is the general reaction. Nucleoside monophosphate kinases. They are specific for a particular base, but not specific for the sugar. So the enzyme can work for, let's say, guanine. This N can be G, but it can be used on ribose form or deoxyribose of the nucleotide. Okay, but in this case, you will see ATP is always used because I said that this is always in excess. The others, GTP, CTP, TTP, no, not as much. You will always see ATP over here. All right. So this is the reaction, and because ATP is used, you will always have one ADP being produced. So if N is G, this becomes ATP plus GMP, you will get ADP and GDP. Okay? All right? Okay? So GTP again, then we'll go through substrate level phosphorylation, etc. Right? Then there's another enzyme called nucleoside diphosphate kinase. So if it's G, C, or T over here, okay, this enzyme can then convert the DP to TP. Okay, this action is reserved for G, C, and T as the acceptor. If the acceptor is ADP, then this happens, okay? This glycolysis process and oxidative phosphorylation process happens for ADP. But if it's C, G, or TDP, you can use any donor group from ATP to make it become TP. Okay? So this reaction is called, catalyzed by nucleoside diphosphate kinase. For example, you have the donor as ATP, right? ATP is the donor donating the extra phosphate, and you have GGP as the acceptor. GGP will become GTP because it has accepted the phosphate from ATP, and ATP will become ADP. Okay, so this kind of passing the parcel game again is reserved for G, C, and T. Okay, which are in general at lower levels than your ATP. Okay? So that's why it also makes sense that there must be another method for making ATP rather than this passing parcel process. Because ATP is required in all these. Okay, so we must make it from another source to ensure the constant supply. Okay? Then we have the salvage pathways. So just now we said we can do it by de novo synthesis, okay, whereby we make it from scratch. But if the nucleotides are broken down in the cells, we can also salvage the free bases by special enzymes like this. Okay? So PRPP is involved again. So PRPP is always used to donate ribose phosphate. If I have DNA broken down because the cell dies, I have adenine floating around. Okay, we can salvage the adenine by adding ribose sugar phosphate, right, onto the adenine to become AMP. Right, so PRPP serves the function. Okay, PRPP serves the function. For guanine, okay, for guanine you have another intermediate called hypoxanthine. Okay, hypoxanthine can be salvaged again by PRPP to become IMP, and then IMP becomes G and C. A and G. And then guanine, okay, floating around can also be salvaged, okay, by PRPP, catalyzed by this reaction, phosphoribosal transferase enzyme to give you GMP. So adenine, okay, purine salvage pathways are common in the body, not pyrimidine. Pyrimidine, the enzymes are not efficient. So usually pyrimidine bases are not quite salvaged in our body. So if you lose it, you lose it. Okay? Alright, so purine on the other hand can be recycled. Right, so our body is really the major recycling, very green mechanism, green machine. Everything is not wasted. Everything is recycled. And finally, just an introduction of how we can inhibit this nucleotide biosynthesis process without discovery. I said that UMP can be converted to C and T, right? C, we have this glutamine added, but in this drug discovery process, there's a way to inhibit U to be converted to T, right? This is because it has been found that in cancer grows quite quickly, right? Proliferates quickly. The UMP to TMP conversion is overdrive, all right? It's in overdrive. TMP is then incorporated into the DNA of the cancer cells. So scientists have found a way to stop this process. Okay, stop this process. But then you know that this is non-specific. This is a normal process, right? If you add this drug here, your normal processes will also be affected. That's why in cancer therapy with 5-fluorouracil, horrible side effects. Right? There are horrible side effects. Very lethal even to the cells, normal cells. But really for cancer, we have no choice. But in these days, it's really about talking about targeted therapy, whereby we target only cancer cells and leave the normal cells alone. Okay, but the old chemotherapeutics like this 5-fluorouracil, it is just indiscriminate. You look at this 5-fluorouracil, it is exactly a uracil molecule. I think I missed out a CO here just now when I draw the uracil. So it is what we call an analog of uracil. It only changes a fluorine added to the molecule. Okay? So in this case, this is FDUMP, this is 5-fluorouracil. Okay? They draw 5-fluorouracil here, called 5-DUMP, and then they put a cross, inhibit. So 5-fluorouracil inhibits this enzyme called thymidylate synthase, which converts UMP to TTMP. Now, UMP to TTMP procedure is a methylation procedure. Okay, so the difference between U and T is a methyl group, right? It's a methyl group over here. The methyl group is donated by tetrahydrofolate. Just now you have N-formyl tetrahydrofolate because the donation is a formyl group. But in this case, it is a CH3 group, no O. Therefore, it is just tetrahydrofolate. Okay? So this whole cycle shows you how tetrahydrofolate gives up its methylene group, methylene, CH3 methylene group, right, to UMP and then gets recycled back. Okay? Catalyzed by different enzymes. The major enzymes that we want to know here is thymidylate synthase. The final step, whereby the methylene group, CH3 group, from tetrahydrofolate is donated to uracil to become DTMP. Okay? The CH3 group is here. Now, if you replace the structure with fluorouracil, this reaction does not happen. Okay? Okay, how does it work? Let's look at this picture over here. Okay? So whenever we draw this shaded blue color is the enzyme. Okay? The enzyme. And the enzyme is thymidylate synthetase. Okay? Thymidylate synthase, sorry. This enzyme is thymidylate synthetase. Enzymes have amino acids on it. Okay? Substrate is the methylene tetrahydro. H form is tetrahydro, 4 hydrogen. Tetrahydrofolate. Okay, it looks something like that. Okay, so our end point is to have this CH3 group extracted and put onto DUMP. So the 2-substrate is being held in the enzyme, and the enzyme has a sulfhydryl group. This could be contributed by a cysteine. Okay, it could be contributed by a cysteine in the amino acid cysteine on the enzyme, and it attacks this carbon over here on the DUMP, right? And once it attacks this carbon over here, it is able, right, to interact with the carbon atom. on the tetrahydrofolate. Okay, right? So this reaction is critical in that sense that the reaction over here, final reaction, is called 1,3 hydride shift, whereby we know that the methyl group should be CH3, but over here we have only 2 hydrogen, CH2, so it's not enough to make a methyl group. So a 1,3 hydride shift occurs to pull this third hydrogen into the first position, okay, to make a CH3. Okay? In order to have this happen, in order to have this hydride shift happen to pull the hydrogen to this carbon, there must be a base amino acid over here. Okay, a basic amino acid over here. Okay, it can be glutamine, asparagine, etc., the basic ones, to help draw and abstract this hydrogen on the carbon. Only when they draw this hydrogen, right, because it's basic, it takes in protons, right? If it's acidic, it cannot take proton anymore. So because it's basic, it draws this proton out, then this CH3 can be attached to this carbon in the final reaction. Okay? So imagine if I have a fluorouracil, if I have a fluorine in this position, fluorine being highly electronegative, this reaction is impossible. Okay? This abstraction of the proton by the base amino acid residue, basic amino acid residue on enzyme will not occur because it's too electronegative. Okay? So once this fluorine is being put here, basically the complex gets stuck. The complex gets stuck. You cannot have a 1,3 hydride shift, and this is what we call a Swiss site inhibitor. The 5-fluorouracil is a Swiss site inhibitor because it causes the Swiss site of the enzyme. Okay, the enzyme basically is stuck in this tetrahedral complex and dead end. Okay, will not be able to function anymore. Enzyme is completely gone. Will have to be degraded. That's why it's so effective. Okay, that's why it's so effective in cancer, not just in cancer, but also in normal cells. Because it's an essential process for cell cycle. Okay, for cells to grow, you need all these nucleotides, right, to replicate your DNA. And when this step is stuck, that's it. No more growth. Okay, so we have come to the end of the purine nucleotide lecture. These are the key concepts. If you put it parallel to each other, you will see a lot of similarities. Of course, with pyrimidine biosynthesis is much simpler. And finally, one way we can inhibit the process. Right, there are many other chemical molecules besides 5-fluorouracil that can interrupt these processes, nucleotide biosynthesis processes, and they're very relevant to cancer. This is an example why. Okay? Because you want to stop cells from growing, stop cells' DNA from replicating. Okay? So we have some time, but I thought maybe we won't go through the fatty acid metabolism because I really want you to go back